What we're going to do in this part of the course is look at some of the environmental proxies that geologists, earth scientists have used in order to reconstruct environmental change over time. And there's various different methodologies available. Some of them are shown here, for example, in these images. This is a website that you can get further information from if you're interested. But in generally, we're going to use the sedimentary, the geochemical, and the paleontological records as our source of data. In summary, there's a bewildering variety of different proxies available. Geologists have been very imaginative in terms of what they have used in order to reconstruct environmental change. So all we can really do is look at some of the general principles that are involved and provide a series of selected examples that illustrate their use. The key guiding principle throughout all of this, however, is that what we do is we take a change over time in some particular variable and we use that to infer a change in some environmental parameter that we're interested in. So, for example, it could be a direct measure of the amount of carbon dioxide trapped in ice over time, for example, and then that would be part of our assessment of what the atmospheric conditions were like. Or it could be something such as the abundance of a particular type of pollen grain that actually then was used to track changes in, for example, the abundance of certain types of shrub or plant. I'm going to start by just illustrating the general principle of what's involved by reference to this particular foraminifera. It rejoices in the name that you see here, and foraminiferas, of course, are either part of the plankton or bottom living benthic. They're microorganisms found in a wide variety of environmental settings throughout the marine realm. They're testate in that they possess a shell which will either be mineralized, as in these examples here, or that's made up of agglutinated grains. And you can see the individual chambers of the shell in these specimens beautifully. They have a series of pseudopodia that emerge from the opening of the shell, and they can often be numerically abundant in any particular assemblage. These are a series of beautifully displayed material collected from a modern beach. They're an important part of sediments that accumulated via pelagic settling out through the water column. So here's a plot that shows the relative abundance of this foraminifera with sea surface temperature. And you can see that in something such as low seawater temperatures that you see here, it can form up to 100% of the assemblage recovered. In other words, the assemblage will be monospecific. And you'll see that the same trend tends to hold whether the temperature is winter or summer. As you actually increase the amount of temperature, the relative abundance of this taxon in the living assemblage decreases. So what we can see then is that this particular taxon dominates assemblages in cold water masses, and when you get to warmer waters, it occurs in much lower frequencies and forms a much smaller part of the abundance of the assemblage. So what you can actually do then is you can take that data and scale it on a global scale and relate it to latitude. And what you find then, of course, is that this assemblage dominates assemblages in transitional through to polar water masses at high latitudes north and south of the equator. And it's very low in terms of its abundance in subtropical and tropical environments. So straight away, even without knowing anything more other than its relative abundance and how common it is, you have a crude measure of where you are on the surface of the globe. One of the interesting features about this particular taxon is that it can actually coil when building its chambered shell either to the left or to the right. And these two images then are for left coiling examples, and the two on the right hand side are for right coiling examples. And what you'll see straight away is that there is a significant difference in the distribution of the left and right forms as you vary temperature and as you change latitude. So for example, the left coiling form dominates in cold water. It can actually be found living in sea ice, whereas the right form shows its peak in terms of its abundance at more intermediate temperatures, neither too hot nor too cold. In terms of the relative abundance with latitude, you can see that the sinistral forms dominate at high latitudes and intermediate latitudes, whereas at lower latitudes it is the dextral coiling form or right coiling form that becomes much more abundant. And finally, just notice that the relative abundance is different between the left and right forms in terms of how important each is in any assemblage. The relative abundance for the left coiling form can be up to 100% of an assemblage, as you see here, whereas the maximum that the right coiling form will be in terms of any individual assemblage will be a relative abundance of about 30 or 35%. One of the key things we would want any environmental proxy we were analysing would be to be able to track that at very, very short time intervals. Ideally, what we would like to be able to do is to track how our environments change at an annual or even a sub-annual basis. And in certain situations, that's actually possible. So for example, of course, if you take tree rings, as you know, these record an annual signal as the tree grows. And that variation in the size of the annual growth rings then can be used as an environmental proxy. The rings themselves are developed in the woody tissue of the tree, the xylem that you see here. And of course it's exposed whenever you take a cross section through the tree. So the xylem in this area here is responsible for transporting water and nutrients upwards through the tree, and the most active part is towards the periphery and towards the center. The heartwood of the tree forms when the cells become inactive and die. On the outside of the xylem you have the cambium, which is the thin layer responsible for the production of new cells, and external to that, the phloem, which transports the photosynthesate downwards from the leaves through the tree. So in other words, the sap, it passes through this layer here. That's the sugar and nutrients dissolved in water that's then redistributed through the tree. Phloem cells that have died are converted to bark and provide external protection for the tree. If we take a little bit more of a closer look at the xylem then itself, at the core of the tree you have the pith, which is a spongy mass with air spaces and cells that store water and starches. Each growth ring can be split into two parts. You have the early wood and then external to that the late wood. The early wood represents typically spring growth. It's light coloured when you look at a cross section through the tree. The cell growth is rapid and the cells are relatively large. That facilitates good water transport. As water becomes more limited during the summer, cell growth slows. You find that denser, darker rings are produced. The tree is undergoing, in this case, a phase of hardening off, and the cell walls will thicken in preparation for winter temperatures. And of course, straight away you can see that there's a sequence of different variables that can be measured. The whole tree ring, for example. So that's a light band and a dark band. You could measure the variation in late wood width, which would then just be coding the dark bands as you move from the core of the tree outwards from older to younger. And then something such as the variation in late wood density would be expressed in terms of how dark one of those dark bands actually was. The other obvious source of high temporal resolution data are varved sediments. The key to retaining temporal resolution in the sedimentary record through these varves is to deposit them in such a way that we do nothing to alter the signal afterwards. So we want to avoid things such as post-depositional disturbance, and we want to create environments in which we have continuous sediment accretion. So we want no episodes where we pause sedimentation and create gaps in our sequence, and we certainly want no episodes where we have erosion and removal of sediment. And we tend to only meet those requirements in very, very specific environmental circumstances. So, for example, if you have what we could colloquially term quiet water conditions, those that lack bottom currents or major intervals of rapid deposition carrying a sediment load that's likely to remove sediment, we want to certainly exclude infauna that will bioturbate the sediment and tend to homogenize it and disrupt any lamination we're producing. So straight away we're thinking of the importance of low oxygen environments or anoxic environments that exclude infauna. And typically we want deeper environments, largely because they're less sensitive to loss of volume of water. For example, if you have a lake that's evaporating, clearly the deeper the lake, the larger the volume of the water, the more likely it is to survive those episodes of evaporation without going entirely dry. One of the easiest ways to stratify your water column to create an environment at depth within it that satisfies these circumstances is to thermally stratify it. And what that does, it introduces density differences with depth, such that you end up with a well-mixed surface layer known as the epilimnion, and that will exchange heat and gases with the atmosphere. You will have then a limited exchange of dissolved matter between the epilimnion and the hypolimnion at depth, which is insulated from exchange with the atmosphere while you maintain this stratification. And in the case of thermal stratification, the gradient that you see is of course temperature, and thus it's known as a thermocline. Of course, if you're raining in particles from above, those will actually be able to pass through the thermocline. Anything that precipitates in the water column, such as carbonate crystals, will pass through. So you will end up then accreting a signal at the bottom of the lake under a protected environment for environmental variables higher up in the water column. Typically, lake stratification is more likely to occur if you have a deeper water body. Typically, if you have relatively shallow lakes that are about 3 to 3.5 meters depth, the wind forces across the surface are usually strong enough to mix the water right from top to bottom, and that inhibits any stratification forming. But if you have deep lakes, the same interval of water may be affected, but you'll have a bottom water that remains unmixed. So the deep lakes, as well as being less sensitive to volume loss, are also more prone to stratify. Critically, what we have in many cases is the episodic or periodic, and there's a difference between those two, breakdown of stratification. Periodic breakdown of stratification would be something that happened regularly and predictably. For example, once a year at a particular season. An episodic breakdown of stratification, that would be something that happened on a more ad hoc basis whenever circumstances were favourable. So you can see here, for example, in this lake in Austria, you have a very pronounced distinction between the epilimnion and the hypolimnion across a thermocline at around November. But by the end of November, you can see that that has actually broken down remarkably. And then as you cycle through into the early part of the year, you see a situation where the thermocline is effectively gone. It then builds up, is at its peak, and then collapses rapidly again. If you have a lake that overturns and homogenizes at least once a year, it's holomictic. Critically, For us, in terms of trying to preserve any sort of sedimentary record from which we can extract an environmental proxy, what we're looking for are meromictic lakes that are, when they experience any circulation, the deep recirculation does not include the entire water body. In other words, there's an interval at the base of the body of water that is not involved in the recirculation in the monomolymnion. So this monomolymnion is excluded from gas exchange with atmosphere for extended periods of time. Underneath that, in the sediment column, is where we're most likely to build up our continuous record of sedimentation with no overprint. The fact that you've excluded this monomolymnion from any gas exchange with the atmosphere means that very rapidly it becomes anoxic typically, and you exclude not just infauna from bioturbating the sediment, but epifauna from living on the sediment. You often get a microbial reduction of the organic matter present, and as a result of that, you can get a buildup of gases such as CO2 and hydrogen sulfide inside the monomolymnion that are trapped there because of the hydrostatic pressure of the overlying water column. Clearly, if you have continual sedimentation, you have a continuous record, and that's most easily achieved by pelagic and hemipelagic sedimentation, where you continually rain material out from the overlying water column. And clearly, if you have a faster sedimentation rate, what that will do is it'll offer more temporal resolution, in that a extended interval of sediment allows the opportunity to sample it, for example, at higher resolution. But the downside is that the volume of your proxy, whatever you're looking for in order to reconstruct the environment, will be diluted in the background material that's accumulating. And I'll just emphasize one point here where you can see quite clearly the impact of phenomena such as bioturbation, where the brown material here has been reworked down into the volcanic ashes. So, signal from this part of the core I'm showing here is actually going to be a hodgepodge from material of this age and also material of younger age that's reworked downwards. And in general terms, bioturbation is one of the key mechanisms by which you will overprint and eliminate the temporal resolution in your cored sample. But in fact, it'll depend on the nature of burrowing, for example, how deep it is, whether the organisms are reworking the sediment or simply creating open structures inside it. And it's also influenced, of course, by the size of the organisms involved and then the rate of sedimentation, because the organisms will move up through the sediment as more sediment is deposited. So the slower rate of sedimentation, the more you would expect a more thorough mixing of sediment to occur. So straight away, for example, what we're able to do is we're able to take something such as a sediment column, wash it, sieve it, extract from it these beautiful little tests of diatoms, identify them at species level. The names here don't matter, nor does the plot. This is just an example to show you. Plot them up against the depth we've extracted them from. And of course, the oldest sediments are going to be at the bottom, the youngest at the top. And hopefully our sedimentation record is continuous without any pauses. And thus we have a continual proxy for environmental change based on how these diatoms change in terms of their relative abundance through the succession. So here, for example, you can see that this taxon has generally increased in its abundance progressively over time. This taxon, on the other hand, is relatively common in the lower half of the core and then seems to collapse in terms of its abundance and become particularly rare, whereas this one behaves in the opposite manner. So straight away, even without going any further, we could make some inferences that there's some sort of environmental event in and around this interval in the succession, and perhaps that's superimposed on some sort of more gradual trend implied by the progressive change that you see here. The key thing, however, is we've no real context in which to put this in the sense that we don't know if this event is happening 100 years ago or 1,000 years ago. So what we need to do is we need some mechanism by which we can translate, as you can see here, from depth to absolute age. In other words, we need some mechanism by which to put our data that we're going to collect as our environmental proxy into a chronological context.